Okay, welcome to your biology paper 2 revision video. We're going to be going through a few different selection of questions from past biology exam papers for paper 2. So feel free to pause the video, have a go at any of these questions, and we'll go through the solutions together. All right, first question then, we've got a graph showing blood glucose level across time. So it tells us that it's important to control the concentration of glucose in the blood. Give one time when the concentration of insulin in the person's blood would be high. Okay, so essentially we need to understand what is the function of insulin. Well, insulin, what it does is it helps to regulate our blood glucose levels, and insulin spikes when the glucose levels in our blood spikes. So when we have lots of glucose in the blood, what that means is we need lots of insulin to regulate it. So in this case, it will be a direct correlation. Whenever we have a lot of blood glucose in our blood, the concentration goes up, you're going to have more insulin. So you can see, you can just read off the graph here. You can give one time, so any time kind of where this peak is, you could just select a time and say that it would be there. So I would say something like 1.7 or something like that would be fine. Okay, next question. Explain the effect a high concentration of insulin has on blood glucose concentration. And so the effect basically is the reverse of what we just explained there. So why would the insulin spike in the first place? We explained because when there's more glucose, you need more insulin to regulate it. So once you have more insulin, what actually happens to the glucose? So it's basically asking us the relationship in reverse. So what you can say is the effect you would get is that it would be lowered, right? So the blood glucose concentration would lower when you have a higher concentration of insulin. And then you need basically another 2 marks to explain why that is. So the idea is that the glucose itself is going to be converted to glycogen by the insulin. Okay, so remember the insulin is going to allow for this conversion to happen, where we take our glucose and we store it into glycogen instead. And the 3rd mark is basically for saying where this happens. So this happens in essentially the cells, or you could also say the liver, or you could even say the muscles. That would all be enough to get you the mark. Okay, so you can see how we structured that 3-mark question. Okay, next one then. In the cold winter months, the echidna, which is basically some creature over here, which we call the echidna, hibernates. During hibernation, the echidna's body temperature decreases to below 5 degrees Celsius, and the echidna sleeps for up to 17 days at a time. The echidna's rate of metabolism slows down. Explain why the decrease in body temperature is an advantage to the echidna during hibernation. So here we just want to think about, why do animals hibernate in the first place, and why would the decrease in body temperature help it to hibernate? Okay, so the reason you want to hibernate is basically to retain your energy and allow all the energy that you have stored in your body to last a really long time. That's the whole idea. When you hibernate, you're trying to basically take out these long winter months. And so what we can write here for the 2 marks, the first thing it says, first thing that we can say is, if we have a low body temperature, we lose less energy. You can say you lose less energy as heat energy from your body. And therefore, the 2nd mark basically to link this to hibernation. Why is this good for hibernation? Well, your body energy store of energy that your body has collected is going to last longer, okay? And you need it to last longer because you're not going to be replenishing it. That's the whole idea, right? You need to link those 2 ideas together. Then the 2nd question linking that then is, explain how the dilation of blood vessels in the skin can help to decrease body temperature. Well, here again, you got to think about what actually happens when you end up dilating blood vessels. So this keyword dilation, what does that mean? That basically is the opposite of constricting. So the blood vessels, they might widen. Okay, so why would we want the blood vessels in the skin to widen, and how can that help decrease the body temperature? Well, essentially, when you widen those blood vessels, what you do is you increase the blood flow. And in particular, because it's in the skin, you increase the blood flow near the surface, okay, or the surface of skin even. But the idea is that you have your blood flowing throughout your body, right? But when your blood is deep within your body, then it's quite far away from the surface. And the reason why you want it to be near the surface is because you can lose more energy like that. If it's within you, then the thermal energy that the blood gives off is going to be absorbed by your body. Whereas if it's towards the surface, you're going to end up losing more of that heat energy. So what you can say here is you lose more heat energy. So more heat lost from body / blood. And then the final point is this whole point is this cools down the blood. So you have cooler blood and therefore a cooler body, okay? Because the blood then flows around your body and cools down that body as well because it's cooler. Okay, let's move on. The athlete's energy intake each day from food was 20,000 kilojoules. The evaporation of 1 centimeter cubed of sweat requires 2.5 kilojoules of energy. 40% of the athlete's daily energy intake was used to evaporate sweat. And I'm just going to go through, underline all the keywords. I recommend you do this in your exam as well. Calculate the volume of sweat the athlete lost each day. Give your answer in dm cubed, and it's given us a nice handy conversion for dm cubed 2 centimeters cubed there. Okay, so let's work through this one together. It is a calculation question, so let's see how you guys do. Okay, so let's work through this one together then. And the first thing that we want to be able to work out is how much of our athlete's energy was used in basically releasing sweat. So we have the total intake was 20,000 kilojoules. That was our food intake. And the idea is you need to use this as your total. And then if you times this by 40%, so to times by 40%, you can times by 40 and divide by 100. And if you do that, then you'll end up with how much energy the athlete actually used to evaporate the sweat. So if you work that out on your calculator, you should get 8,000 kilojoules. So now we know that there was 8,000 kilojoules of energy that was used to evaporate that sweat. And we're told that 1 centimeter cubed of sweat requires 2.5 kilojoules of energy. So if we have 8,000 kilojoules, and how many centimeters cubed of sweat did we release? So we can do 8,000 divided by how much energy 1 centimeter cubed takes, which is 2.5 kilojoules. And they're both in kilojoules, so we can ignore the units for now. And then you should be able to get the amount of sweat that was produced in centimeters cubed. And that is 3,200 centimeters cubed. And the last thing we want to do is convert this to dm cubed. And so it's given us a conversion over there. It's just divide through by 1,000. So you end up with 3.2 dm cubed or decimeters cubed. Okay, and that would be your final answer. Oh, it kind of went off the screen, but there you go. Okay, then the first part of the DNA questions now. So the question will ask if the nucleus of a cell contains DNA. Name the structure inside the cell nucleus that contains that DNA. So it's just about cell structure here. You either know it or you don't really, and the structure inside the cell itself is going to be the chromosome. Okay, so the chromosomes are what contains that genetic information and that nucleus. So the structure inside the nucleus itself. Okay, so the nucleus contains the DNA, but within the nucleus you have chromosomes. Okay, the second part then we got a nice diagram. Figure 1 shows part of a DNA molecule. Name the part of the DNA molecule labeled X. So this is this over here, this kind of pentagonal shape. So I'll let you guys have a go. What do you think this X is? Well, it is actually a sugar. Okay, now you don't need to actually say the specific sugar that's involved in DNA. If you say it's a sugar, it's fine, but you should know the general structure of DNA, where you have the sugar backbone. Okay, so technically DNA is deoxyribonucleic acid. So if you were to say deoxyribose, you would also get the mark. Okay, if you were to say ribose, that's not correct, but it is a sugar. So as long as you identify that, that's fine. Next, we've got a couple questions. What type of substances are A, T, C, and G? And what do they each stand for? So I'll let you guys have a quick think about this one. So the parts labeled A, T, C, and G, these are what we called bases. Okay, so DNA bases. That's the type of substance they are. And what do they each stand for? So they stand for the A is for adenine, the C is for cytosine, G is for guanine, and T is for thymine. Okay, so that's, you should know that, hopefully. The next question we've got, a cell sample was taken from John's body. It was found to contain 4 times 10 to the minus 12 grams of DNA. How much DNA would a human sperm cell contain? So what basically this question is asking you to find the link between a regular body cell and a sperm cell, or a sex cell specifically. So I'll give you guys a chance to have a go at this one too. And the key part here is that the body cell had 4 times 10 to the minus 12 grams of DNA. Okay, if that is the case, then essentially how much DNA would a human sperm cell contain? Well, it's basically got exactly half the amount of DNA. So remember, the sperm cell combines with the egg cell to form a complete cell, if you like, with the complete genetic information. And so in the initial stage, the sperm and the sex cell only has half of the expected DNA, or half of the DNA of a full adult human cell, body cell. Okay, so then we would just divide that by 2 to get 2 times 10 to the minus 12 grams of DNA. Okay, now we're moving on to some plant questions. So the growth of daisy plants on a lawn is affected by biotic factors and by abiotic factors. Table 2 shows 6 factors, and you want to basically sort which ones are biotic and which ones are abiotic. Okay, so I'll give you guys a chance to go through those. Okay, so let's go through this one. So we need to know the difference between a biotic factor and an abiotic factor. So a biotic factor is basically is a factor that affects the growth, but it's dependent on a living thing, a living organism that might reproduce. Basically, anything that's dependent on a non-living organism, such as habitat conditions, chemicals, or things like that, would all be abiotic factors. So with that knowledge, then we should be able to sort these out. So nitrates in the soil, well, nitrates are not a living thing, so these would be an abiotic factor. Rabbits eating the plants, rabbits are a living thing, so that would be a biotic factor. Shading by a building, again, it's not a living thing, so it'd be an abiotic factor. Soil pH is also the chemical makeup of the soil, also an abiotic factor, it's not a living thing. Temperature, again, an abiotic factor of the environment, not by a living thing. And trampling by people are living things, so that would be a biotic factor. Okay, now we're given this figure which shows a food web. So a food web is basically like a food chain, but it's made up of lots of different food chains. You can see that it's kind of interconnected. Okay, so there's a food web for you guys. The animals in figure 9 get their energy by eating other organisms. Describe how algae get energy. So over here we've got some algae, which I will circle. And the question is, okay, the algae is not eating anything, right? It's going into these different animals, but there's nothing going into the algae. So where does the algae get its energy from? So I'll give you guys a chance to have a go at that. And essentially the algae are able to get their energy from light, or it would normally be from sunlight specifically. Okay, so that's fine. Then it's asked us to name one producer and one primary consumer from our food web. So if you guys have a look at this and tell me, can you spot one producer and one primary consumer? Well, hopefully everyone got this right. It's not too complicated, I don't think. The first one, the algae, you can say is a producer, since it's producing, it's the first source, if you like. And then a primary consumer is basically the next level up from the producer. Okay, so you could say krill, or you could even say a copepod, or the other one you could say is a herring. Okay, because all of those, they feed directly off the producer, so they would be the one level up, the primary consumer. Okay, let's move on. Next, we have this genetic question. So we've got this diagram basically, and we've got the key for this diagram. Basically, this question is about sickle cell anemia. So you can see in our diagram is keyed differently. If you have an affected male, an unaffected male, a male with sickle cell anemia, or a male with a sickle cell trait. So what does that mean? We've got some more information down here. Essentially, if you guys should be familiar with sickle cell anemia, you have the allele, and it's only if you have a if you have a homozygous mutated allele, then you end up with sickle cell anemia. Okay, so it's kind of listed out here, and we'll go through the information actually just to make it a bit easier. So a person who's homozygous for the normal hemoglobin allele produces normal red blood cells. Now, you guys should know what homozygous and heterozygous is, so I'm not going to define it just yet. A person who's homozygous for the mutated alleles, and also alleles you should know the meaning of, produces red blood cells with abnormal hemoglobin, and has red blood cells that can form an altered shape, and has sickle cell anemia and becomes ill. Okay, that's the key thing. If they have homozygous mutated alleles, then they have sickle cell anemia. If they have homozygous normal alleles, then they have normal red blood cells and they don't have it. A person who is heterozygous, so this is the interesting one, has both normal and abnormal hemoglobin in the red blood cells, and has sickle cell trait, is generally healthy but can become ill in certain circumstances. Okay, so then we go to our question itself. Determine the probability that the child will be a girl with sickle cell trait. And so the persons 8 and 9 basically are expecting a second child, and so we need to give this probability. Okay, and to do it, it kind of gives us some tips on how to do it. And so I will let you guys go ahead and have a go at this question. And there's the diagram for you if you want to pause it on here. And remember, the question we're asking is number 8 and 9 are having this child, number 13. You want to give the chance of that child being a girl and having sickle cell anemia. Okay, so go ahead and have a go. All right, so there was a lot of words in that question, but actually, when it comes down to it, the important thing is just to understand what they're actually talking about. Okay, so the homozygous and the heterozygous alleles. So the alleles are the different forms of our phenotype. Okay, so identify the phenotype of each offspring. Sorry, identify the phenotype of each offspring genotype. Okay, so that's what we want to do, and we want to use these symbols HA and HS to represent that, and we'll draw a Punnett square diagram at the end as well. Okay, so let's actually go through it. So the first thing is we have number 8. Now, if we look at our key, number 8 is an unaffected male, and if you scroll down to an information, the unaffected male is the people who have homozygous alleles HA. Okay, so this person basically has HA. So we can actually add that into our diagram just to keep track. So this number 8 is going to be HA. Okay, perfect. And then the next thing is number 9. So number 9 over here is going to be a female with the sickle cell trait. And so what does that mean? Well, female with the sickle cell trait, if you go down over here, person who is heterozygous has both normal and normal hemoglobin in the red blood cells and has a sickle cell trait. Okay, so basically what that means is if they're heterozygous, they have one of each type. Okay, so they have a HA and they also have a HS. So now the question is, they have this child number 13, and we want to know number 13, what are the chances they're going to be a girl with, sorry, it's not number 13. Number 13 is a child they already have, but they're going to have a second child. So we want to say, what are the chances that this child will be a girl with the sickle cell trait? Okay, so we can solve this now. So if we draw out our Punnett square, it should become pretty clear to us. So the first thing, we can put the father, which is the male, on the top. So he has HA, and we can put the mother on the side. So she has HA, HS. And then we can draw a Punnett square like this. And so when you have HA, you're just going to get HA in the middle, and the same thing is going to happen here. And then you're going to have HA, HS over here, and then finally HA, HS over here as well. Okay, so this basically tells us the different combinations of the alleles that we'd get. And so remember, the ones that we're interested in is if the girl has the sickle cell trait. So if she has a sickle cell trait, that means she has heterozygous alleles and she has both the HS and the HA. Okay, so basically we can look through our Punnett square to see how often that happens. So you can see it happens here and it happens here. So 2 out of 4 times, 2 out of 4 of the combinations, you're going to end up with the sickle cell trait. Okay, so the chance here that we worked out was 50%. But here you need to read the question a little bit carefully, because it does say what are the chances the child will be a girl with the sickle cell trait. So in the first place, the chance of having a girl is 50% as well. Okay, so a 50% chance of having the sickle cell trait, but there's also going to be a 50% chance that it's a girl. So you could have the sickle cell trait, but it's a boy, right? In which case, that's not answering our question. So to work out the basics, the combinational probability of both of them, you need to combine it. So it's basically 50% times 50%, or 0.5 times by 0.5 to get the total probability of it both being a girl and that girl having the sickle cell trait. And so the end result you should get, it will be a 25% chance, or 0.25, or 1 in 4, something like that. That you just have to write down the probability. Okay, so a lot of people, they miss out this last step, but if you read the question carefully, then hopefully you did get that right. All right, so we'll leave it there then for biology paper 2. Hopefully that video was useful. If it was useful, please do leave a like, do subscribe if you're new to the channel, and I'll see you for the next one.